Hello everyone, I'm Shelly and I'll be presenting my talk on immune modulation and 3D co-culture models, paradigm shift in regenerative endodontics. Before I begin, I would like to thank POPMA and specifically Dr. Bhuvan for giving me this opportunity. And before we dive deep into this topic, which is close to my heart as it's related to my PhD and further my area of research interest, which I'm doing these days, I would like to thank to my alma mater for equipping me with this knowledge and giving me these opportunities so I'm able to speak to all of you today. So my BDS is from Manipal and my MDS from Bapuji Dental College Hospital, Davangere, and my PhD is from University of Otago, New Zealand. And this is the place where I work at School of Dentistry, IMU University, Kuala Lumpur. Okay, so as we're going further into the game, the topic is immune modulation. So I'm divided my topic into the brief introduction. I'll give you the brief glimpse of what is immune modulation and how it works for the dental pulp therapy. There are various roles of immune modulation. Those various kind of means the molecules, those biomolecules, those microRNAs, those anti-cytokine antibody, they all are using this immune modulation to regulate dental pulp inflammation. So, but we will be specifically seeing how these anti-cytokine inhibitor or antibody is used. Then I'll give you the how to differentiate between two-dimensional and three-dimensional cell culture models. What are the challenges and the future direction in this approach, and take-home message. So, what is dental pulp? We all know dental pulp. We have been reading from our undergraduates from the BDS. It is a specialized connective tissue which consists of odontoblast, fibroblast, immune, and progenitor cells. Now, we all have read that they have mesenchymal cells, those wandering, and the magical cells are dental pulp stem cells. We all know that the mesenchymal, they are pluripotent stem cells. Now, these cells, they are very promising for cell-based therapy. And these are the cells which are helping to the pulp, help in the repairing of the pulp as well. And with all this, what is forms a broader heading called it the regenerative endodontic procedures, which enhance the pulp repair or pulp healing or pulp regeneration. Now, what is immune modulation? Now, immune modulation is the process of altering or regulating the immune system activity to achieve a desired therapeutic effect. Now, this approach is constantly used, like immunotherapy. It is constantly used in cancers. Most of the cancers, they are using it like personalized medicine. So, same, this therapy is now being, it's again in very infancy stage. It's being used in the pulpal diseases. So whatever the immune modulation is being used, it's only in the in vitro or maybe the animal studies. The clinical trials are lacking. But this immune modulation just does not imply suppressing the immunity, so rather regulating the immune response. For example, it reduces the harmful effect of inflammation. At the same time, it initiate the healing. Okay, it initiate because a little bit of that the inflammation and regeneration goes hand in hand. Little bit of inflammation is required to initiate the process of healing. Okay, so both goes hand in hand. Okay, so as I told you, now this immune modulation or this therapy, excuse me, I'm just trying to move the cursor up. Modulation for the dental pulp therapy, there are various kinds of molecules that are used, like molecular therapy, using adrenomodulin molecules, cell therapy, including mesenchymal. Now, but we are going to focus on anti-cytokine and antibody molecules. Now, when you're going to use this anti-cytokine antibody molecule, I would just like to give you a brief introduction that why we are using or focusing on this molecule. Various chronic inflammatory diseases such as psoriasis, inflammatory diseases like inflammatory bowel disease, rheumatoid arthritis, they're characterized by dysregulated immune response and relatively high cytokine level. Now, this interleukin 23 antibody have been successfully used in these chronic inflammatory diseases, and a very good response has come. Okay, so similarly, the pulp inflammatory disease or pulpitis is also characterized with a relatively high cytokine level. But and this approach has been tested in the chronic inflammatory diseases and it blocks the specific inflammation pathway. But this approach is not yet tested in cases of pulp inflammation. So, with this background, that was the research of my PhD. That the study aimed to assess the effect of interleukin 23 blockade or interleukin 23 inhibitor as a pulp immune modulating agent in vitro and in vivo. So the hypothesis of my research was that interleukin 23 inhibitor modulates pulp inflammation and facilitates pulp healing both in vitro and in vivo. So the specific objective of my study were to establish the primary culture of non-carious and carious human dental cells. And then we compared, once these cells have been grown in the lab, we try to characterize them and we look for certain characteristics like their proliferative potential, their self-replicative potential by measuring their colony forming efficiency, their differentiation potential by looking into their mineralization potential and looking at some molecular expression by doing their gene analysis. And next objective was to compare the effect and addition of by the addition or when the cells were exposed to interleukin 23 inhibitor. So what happened to these cells, their growth, their viability. And once it was done in vitro, the same drug, that is this interleukin 23 inhibitor, was tested in vivo model, that is in this Wister rat on their mandibula first molar. So this how I established the cell culture. So the primary cell culture was established from the freshly extracted third molar tooth. As soon as the tooth was extracted, just I'm going to tell you in very brief, it was extracted, the pulp was taken out, it was harvested, it was cut into small pieces, plated in this 6-well culture plate, top up with the cell growth media, and kept into the incubator. So what happened now is the magic. So after almost a period of 7 days, you can see this is the pulp tissue. Now this picture is under the microscope, and to look into the cell culture, we use inverted microscope. So this is the pulp tissue, and you can see the cells coming out of it. So this is the pulp explant, also called as mother explant, and you can see these dental pulp cells are coming out. So same dental pulp explant. Okay, now this is at 7 days. You can see more cells are coming out, and at 16 days, a lot of cells are there. Almost the culture plate is filled. So when this stage comes up, so the next what we do, we transfer, we we passage the cells. That is, we move the cells from the smaller vessel to a larger vessel. This process of moving the cells, we dissolve them with another substance called trypsin, that the trypsinization is done. And then we move the cell because we need lots of cells for doing the assays. Assays means the experiments. You all know that. Okay, so now you can see those were passage 1, these are passage 3 cells. Now they are nothing but looks like the fibroblast like cell because they are human dental pulp cells. Again, if you notice, I'm not saying that I have done the experiments using dental pulp stem cell. What I used is the human dental pulp cells, because if we just restrict ourselves to the stem cell, we need to do some sorting procedure, because we know human dental pulp cells has various type of cells in it. Fibroblasts, the progenitor cells, these mesenchymal stem cells like the dental pulp stem cells. It has endothelial cells, immune cells, nerve cells. So I've not done any kind of sorting procedure. So that's why I'll be using the term human dental pulp cells. Now, once this all the cell culture have been established, that is the carious as well as non-carious. Non-carious means healthy, and the carious are the inflamed one. So further, as I told you during when I was mentioning about the objective, that all these assays were done to further characterize them. The immunocytochemistry was done to look for the mesenchymal expression, then to look their growth kinetics, to look at their self-replicative potential. Now, another essential ability of these dental pulp cell, if they have those dental pulp stem cell population, is ability to differentiate into a mineralizing phenotype that is confirmed by mineralization assay, that is alizarin red assay was done. The gene expression analysis is done. See what are the different type of genes expressed in the healthy and in the inflamed dental pulp cell. And this MTS assay was done, cytotoxic assay, to find the correct percentage, like I mean, when their cells were exposed to the different type percentages of interleukin antibody, that is interleukin 23 antibody. So which is the right percentage at which the cells are viable, they are showing maximum viability, they are able to proliferate. So to measure that, MTS assay was done. Okay, so now some very interesting results came for this MTS assay. So cells were exposed to the different concentration of interleukin 23 inhibitor, that is 1.25, 2.55, and 10 microgram per ml. So the measurement, the cells were exposed for 3 days, for day 1 and day 3. So if you look at this bar graph, so the blue one represents the normal cells, means the healthy one, and these are the inflamed, the red one. So if you see at day 1, the viability of the healthy cell is decreasing. So it means they were not able to tolerate the higher concentration of interleukin 23. But however, the inflamed dental pulp cells were surviving. They were showing 100 plus cell viability, and the results were found to be statistically significant. Similarly, after day 3, or the results were again amazing, especially at higher percentage, including 2.55 and 10 microgram per ml. The inflamed dental pulp cells were able to thrive. They were showing good viability. They're showing 100 plus viability, whereas those in normal dental pulp cells were not able to survive those higher percentage of antibody. So it means some kind of modulation happens, some blockade happened in carious dental pulp cells, and cells were able to grow. So moving further, so once we found that this is the right percentage, we did some experiment in the animal model, that is in the Wister rat. So we took almost like 12 rats for this assay. So group A was the control, where we use saline as a dressing, and group B, we use this antibody. So teeny tiny pulp was exposed. Now it looks very simple, but this experiment is really, really difficult. First of all, the ethical issues, and working with rats is not easy. And because their molar is 50 times smaller than our tooth, that means like our molar. So preparing those, I mean, so this is, it was really very challenging for me to do this, but however, I did it. Okay. So you can see here a small pulpotomy was this, a pulp exposure was done. So this is a pulp exposure. Okay, so the minimal pulp exposure was created. Once it was done, the bleeding was controlled, and with the paper points and slightly. Once the bleeding was controlled, the control group received saline as the dressing, and the experimental group received the antibody. And after they were the dressing was given to them, they were restored with GC-Fuji9. Once this was done, the Wister rats, they were euthanized at day 1, day 15, and day 30. They were time points. And when they were euthanized, their jaws were extracted and harvested. They were decalcified using EDTA, and which almost took 2 months to decalcify their jaws. And we got some very amazing results here as well. So at day 1, you can see that day 1, both the groups showed the similar kind of pulp inflammatory reaction. Like there's inflammation, there is pulp hyperemia. But at day 15, if you see, this is the control group, you can still see necrosis and inflammation. But in the experimental group, we saw the mineralized tissue formation. There is inflammation, but there is mineralization going on as well. But at day 30, there is inflammation, there's still necrosis, whereas in the experimental group, the pulp is viable, there's a mineralized tissue formation. So with this, we concluded that interleukin 23 blockade has the potential to promote mineralization in a rat exposure model. And further, it may be promising like interleukin 23, has the potential to limit pulp inflammation and facilitate healing for regenerative endodontics. So currently, this project, my supervisor is still continuing it, and they are at RCT these days. So the clinical experiments are going on. Means the clinical trial is going on, and still at University of Otago. Okay. Now, with this story, I'll move ahead further. What are two-dimensional cell culture model? Now, when I told you that I grew the cell on those cell culture plates, so basically, most of the studies in the literature, you will see that they are two-dimensional cell culture model, which basically means, so this is a fundamental cell-based approach. So basically, it means the cells, they are grown on a flat surface. Okay, if you see here, this is a monolayer of cells, the culture vessel. So the cells are growing. So that's what the two-dimensional means. So they're growing in one plane, that's in the horizontal plane. It is easy to establish and maintain, they're cost effective. But what is, however, they cannot recapitulate the structural organization. So now, for example, in our tooth, the cells, they're not growing in one direction, multi-directional. It's the three-dimensional, right? So they're not only growing horizontally, they're growing vertically also. Now, cell-to-cell interaction is not possible in case of 2D cell culture model. It may be there, but the cues are not there, especially in two-dimensional cell culture models. So they do not capture the crosstalk. So that's why we are moving to three-dimensional cell culture models. Now, what does this, so what does this three-dimensional cell culture models? As I told you, so there are some limitation in two-dimensional cell culture models, as the cells, they're just going in one direction. So we need these kind of cell culture models. Okay, but however, they're technique sensitive, they are expensive, right? You need specific skills for that. Okay. But another good point is that they simulate the bodily environment. Okay, so they have that ability. Okay, so what are these? What are the various type of 3D cell culture model? Now it includes organoid based model, as you can see in this picture. They can be in hydrogels. They can be interconnected porous scaffolds like 3D bioprinted models and microfluidic devices. Now, they more accurately replicate the range of complex cell and molecular interactions. Now, I think you might have heard of organ on chip and tooth on a chip, which I will tell you in coming subsequent slides. Now, just to give you another example, now this of the study is done by one of my very good friends, and he's from UQ, University of Queensland. So they have done this beautiful study. Just trying to tell you how the scaffold is now. They have grown biofilms and not cells, but this concept remains the same about 2D and 3D scaffold or substrate. So it's a 2D substrate, and this is the 3D substrate they have used. Now, essentially in the 2D substrate, as you can see in the picture, the bacteria, they're growing in the one pattern, horizontal pattern, but in the 3D, they're growing both horizontally and vertically. This is just to explain you here. I'm not showing cells here, but of course, the other studies might be there, but I found this illustration best, so I just thought of sharing with you guys. Now, what are the various biomaterials used in this scaffold-based approach? Now, this I have taken from this article of Dr. Warren. So the various scaffold-based approaches that are used, like what are the materials used? So it's the polymers, which can be synthetic and natural. The table, this is a table given in this of the article. It's huge, so I'm just put just one example. It can be polylactic acid, it can be PLGA or polyethylene glycol, and their natural polymers can be used like chitosan, collagen, gelatin. Then hydrogels are there, again collagen, they're host-derived scaffold like platelet-rich plasma. So biomaterials, I mean, that lots of biomaterials that are used, and lots of research is also going on, and specifically just for which type of biomaterial can be used. Because, you know, this scaffold, essentially, it provides extracellular matrix for the cells. So the cells can attach themselves, can grow and proliferate. So that's what simulate the natural pulp environment. So the natural pulp microenvironment can be simulated using these approaches. So again, trying to just recapture what I've covered. 2D. So it's essentially the cells growing on a flat surface, and the 3D in the cells they're growing in horizontally as well as vertically. So they're growing in a three-dimensional environment, which simulates the bodily or the pulp microenvironment. Now, there are other three-dimensional, specifically three-dimensional co-culture models. These have been established. There's something called as Dent-Cyto tool. So what is there in this tool? What they've tried, this Hartzikus et al., what they've tried to uh, model a pulp analog, dentine pulp analog. So as you see, it has two compartments, the upper compartment and the lower compartment. Now this upper compartment is modeling or simulating the dentine. So what they have done is they have grown the cells here from the dental pulp stem cells. They have grown odontoblast-like cells, and then they are placed onto this microporous membrane here. And further, they put the treated dentine matrix here. So this simulates the dentine. And in the lower compartment, what they have done, they have used a co-culture of endothelial cells and the scabs. Scabs are the stem cells from apical papilla in a hydrogel. Hydrogel is the scaffold. So this resembles the dental pulp analog. And now another interesting finding is what the authors have found in their study, they have mentioned that they have exposed this in vitro model, this 3D model, to either the resins and LPS, and they really found good results which are matching the clinical scenario. Okay. There's another study which I mentioned about the using of human 3D dental pulp organoid model. Now, these models essentially in their study, the authors have mentioned they have been used to screen dental material biocompatibility on drugs. So this organoid in this study is composed as a co-culture of human dental pulp cells and endothelial cells. So another interesting finding, what they found is after exposing this organoid to LPS, we know LPS is lipopolysaccharide, the bacterial component. So this is what happened during the caries as the bacteria and the bacterial toxins that reaches the pulp, there's inflammation. Similarly, so after exposing this organoid to LPS, they found that the high levels of interleukin-6, that's the inflammation. So it's simulating the bodily environment. So this is the result shown by them. And similarly, that's what I told you, tooth on a chip or organ on a chip. These are microfluidic device. Okay. The size of this device is just like a USD. And this study is done by Franca Group et al. So this is a device. It is comprised of, if you see here, they have two parallel chambers here. And it is separated by a dentine. Now, this model essentially it mimics the tooth cavity, right? So where the dental pulp interfaces directly with the dentine, and again it is indirectly continuing with the oral cavity. So authors have mentioned in this study that it enables the quantification of responses of pulp cells to dental material and bacterial products. I can explain about this model in detail, but I think that you guys won't be able to too much of the theory. It's a microfluidic device, they can do many things in this together in this chip, basically. Okay. So as we are moving forward, we are moving from 2D to 3D, from 3D to 3D co-culture model. So co-culture essentially means it's just not only just growing the cells together, it's like to have the cell interaction. It can be the culture of the bacteria or of the cells as well. So there are challenges and their future directions. Okay. So first of the thing that needs to be done, like as of now, there's so many proposed in vitro or the lab study. We need to transfer them. Okay, translate them. Okay, to the clinics. Then we need to do some kind of a standardization, or maybe again the personalized medicine is needing, or as does the one size will fit all? We don't know because the inflammation of the pulp, if we see clinically, it varies from individual to individual, right? And another thing which is lacking is the clinical trials, as I've said. So in regenerative endodontics, there are very few clinical trials done. So I think there are big groups which are working on it. They may be We need to do more of clinical trials to come up with some findings. So the take-home message from this presentation of mine is that immune modulation is central to pulp regeneration. And 3D co-culture models, they're powerful tools for research, especially in the area of regenerative endodontics. And as we're moving ahead, we need more of these biology-based therapy. And I presented a part of my research in Taiwan in 2023, and I got the best oral presentation. Just this is the picture I like to post with my co-supervisor, Prof. Ellison. And this would not have been possible without the support of my amazing supervisory team, which I'm in touch, still touch with them. And I hope I'm able to share a little bit of my knowledge with you. And thank you for your time and listening to me. And I'm happy to answer your questions. Thank you.